the endoplasmic reticulum, I like to call it the subway system, and basically it carries products, forming them into vesicles, which basically are phospholipid membrane-bound boats, and they carry them to the Golgi apparatus. But we do have the two different types of endoplasmic reticulum. We have the rough, and of course we call that rough if it has ribosomes on it. So the products that it's going to build are going to be protein-based products, whereas the smooth endoplasmic reticulum is not going to build proteins because it doesn't have ribosomes, but it is going to help in building different types of cholesterol-based products. It's going to help in breaking down different non-protein products. It's going to help in fat metabolism, and it even helps in detoxification of different substances, including drugs. The Golgi apparatus, again, is like a factory where we finish building our product, or almost finish building our product. And so what we do is we take the structures that we started building from the endoplasmic reticulum, we fuse the vesicle that comes from the endoplasmic reticulum to the Golgi apparatus, where we release their product into the Golgi apparatus. And then the Golgi apparatus is full of all kinds of different enzymes, and basically it allows different chemical processes to help complete or almost complete the product, and then it usually will pinch off a vesicle from it, and then that vesicle will usually do one of three major things. It'll either secrete the product somewhere else, it will actually incorporate things into the cell membrane itself, or it will even form specific organelles called lysosomes, which are basically full of digestive enzymes. Here we see a great example of just what the Golgi apparatus is doing, and really part of what the cell does overall, which is create different products, build different products. The cell builds most of its own proteins. The cell builds most of its own structures. After that it needs to replace damaged structures. So if you'll notice, we have the blue, in this picture, the blue rough endoplasmic reticulum, the ends of the endoplasmic reticulum called cisterna, and the ends usually pinch off, carrying the product that was made at the ribosome in a transport vesicle, and then the transport vesicle moves to the Golgi apparatus, fuses with it, releases the product into the Golgi apparatus. The Golgi apparatus is full of enzymes, which are again going to modify and package different products that are being made. It's going to then pinch off the product within another transport vesicle, and then that's going to take one of three pathways. Either it's something that's going to be contained in the cell, or something that's going to go through exocytosis and release to other parts of the body, or if we look at pathway 2, we see that there was something created that needs to be incorporated into the cell's own membrane, and there are many different reasons why that could happen, whether it's either putting some sort of protein channel into the cell membrane, or whether it's repairing the cell membrane with phospholipids. There's so many different things it can do, but the cell membrane is very dynamic. It's changing all the time, and this is how we put products into the cell membrane, the plasma membrane. And then finally we actually see a vesicle that is being released, and that is basically a lysosome because it has digestive enzymes in it, and those digestive enzymes do many different jobs, anything from helping break down injured or dying cell parts to helping unzip packages that have come into the cell from outside so we can release the package or modify the package in some way. Lysosomes extremely important. This could also be something like what a white blood cell would do to help destroy what it engulfed. Again, we see lysosomes here, and we understand that they're full of usually digestive enzymes, things that are breaking things down, one of those types of chemical reactions that occur within the cells. We also have perioxysomes, and they're similar to lysosomes, but instead of having the digestive enzymes, they usually have oxidase enzymes. Basically, they have hydrogen peroxide inside of them, and so they release oxygen, and this helps break down free radicals like hydrogen ions or other free radicals that are very reactive chemicals that would be pulling stuff away and breaking down other elements in the cell. But the peroxisomes will actually help break them down, and also just detoxify different things that might be brought into the cell. And then, of course, they do replicate by actually pinching in half themselves. You know, the mitochondria performs one of the most important jobs of the cell, and that is to basically be an energy converter to help take a carbohydrate such as glucose and break it down to the point where we develop this other source of energy that's appropriate for cellular use called adenosine triphosphate. Now, we can actually have ATP production without mitochondria, but we get a tremendous amount more of ATP when we use mitochondria. And again, mitochondria is using the breakdown products of food, particularly glucose, but it can use fatty acids, it can even use amino acids if it needs to. But the other component that it needs besides the energy source is oxygen. And then the last thing, that reason why it occurs so well in a mitochondria, is because it is full of enzymes called ATP synthase, which tells us that their whole job is to synthesize or make ATP. So mitochondria, cells that are extremely active, that are doing a lot, are going to have a lot of mitochondria. Now, a red blood cell is not going to have mitochondria, and the main reason is because mitochondria uses oxygen, and the job of a red blood cell would be to deliver oxygen, so it would be counterproductive. Now, red blood cell still has need for ATP energy, but it gets it by a process called glycolysis, which we will talk about later. But most cells have mitochondria, and cells that are active, such as muscles or neurons, are going to have a tremendous amount of mitochondria. The cytoskeleton is basically the framework of the cell, and it basically is kind of like the skeleton of the cell itself. It is usually protein fibers and structures that kind of hold the cell in place. A lot of organelles are actually attached to it, and sometimes when those protein fibers grow or shrink or move in some way, then they will move the organelles from one area to the other. So the cytoskeleton is important. We know that also the proteins within a phospholipid membrane, often there are certain proteins that actually attach to the cytoskeleton, so it can anchor that membrane to the rest of the cell. So it holds the cell structure. It actually attaches to organelles, it attaches to the membrane. It actually can move things around. Centrioles are, again, these protein structures that are necessary for cells to be able to divide, because centrioles pull spindle fibers across chromosomes attaching to them, so they're necessary for mitosis and cell division. One interesting thing is that cancer being this, meaning that cells are going through cell division when they're not supposed to, we know that certain cells in the body that either lose their centrioles or don't have them at certain parts of their life cycle don't have the ability to divide. Therefore, they don't have the ability to become cancers. So the majority of neurons do not have centrioles, so they can't divide. Whereas the glial cells, the neuroglial cells, they do have centrioles. So when we hear of somebody having a brain tumor or brain cancer, it's really not cancer of the neurons as much as it's cancer of the support cells that actually have centrioles. Of course, as we started this whole chapter, we said that there's something called that doesn't really exist that we're going to study called a general cell. But here we see some more. Here again, we of course, as we started this whole chapter, we said that there's something called that doesn't really exist that we're going to study called a general cell, but in reality, cells are very diverse. There are a lot of different cells. Now, they are living structures. They do have these three parts. They do have these organelles for the most part, and but they do different jobs in the body, so they don't always express the same genes or the same proteins or the same exact structures. Matter of fact, some of them even lose some of the structures in their development. In our picture, we see some fibroblasts, which are very important cells within our connective tissue structure. One of their main jobs is to produce some of the protein fibers that are in the matrix. And so they have a specific job. They're very common. They can actually even turn into different types of cells. But they're very, very important for our connective tissue, especially the fibrous connective tissue. We see erythrocytes, which are also connective tissue cells, but they're our red blood cells, and of course they're very specific for a specific job of carrying oxygen to all of our cells. So actually we have more erythrocytes than any other type of cell. But notice there's no nucleus and not many organelles inside. And at one time in their development, they actually had both a nucleus and organelles. But they were triggered to produce a bunch of a protein called hemoglobin, and that basically crowded out the nucleus and most of the organelles. So they only have a life cycle of about 120 days, but they do a very specific job. Now, if you look down, you'll notice that we have some epithelial cells, and of course, our epithelial cells are linings, and they make up our glands. They're used to absorb and secrete things. They're used to protect things. And they're very tightly packed cells with a free end on one side and usually a basement membrane covering something else on the other side. So we talked about them during our tissues. Here again we see a couple of our muscle tissue cells. We see the skeletal muscle cell, which of course is what we would find in our skeletal muscle, and long cylinders, multinucleate, the contractile filaments in there. We actually see little units called sarcomeres that have the actin and myosin. Again, we'll be talking about those things in upcoming lectures. And we see our smooth muscle, which primarily lines our internal organs, and notice that they are single nucleate and they're kind of tapered, spindle-shaped muscles. But of course, what muscle basically does is it shortens or contracts, therefore it moves things. Here we see some more diversity, everything from something as an adipocyte or fat cell. And of course, if you'll notice the structure of the cell, the majority of the cell is used to do what the cell's purpose is, and that is to store triglycerides or lipid droplets. So we still have the other parts of the cell. It's just that we don't have a lot of the organelles or in large amounts. We don't need a lot of mitochondria. We don't need a lot of lysosomes. We need storage area, but it's still a living cell, so we need to function as a living cell. Therefore, we are going to have the cytoplasm and the organelles. But the cell's going to be doing a specific job, so the majority of the cell is going to be dedicated to that job. Now, if we look at our macrophage, which is a white blood cell, its main job is to engulf or phagocytize different invaders in the body, such as bacteria or viruses or other invaders, other possible invaders. But when it engulfs it or phagocytizes it, and it does that by kind of using its pseudopods, which is almost like leg-like extensions of its cytoplasm, it actually engulfs, surrounds, and pulls in through endocytosis the invader, and then it needs to one of the ways it kills them is by releasing lysosomes, and there are some other ways it does too, but we can see in this case there are several lysosomes in here because they have these digestive enzymes that helps destroy what it actually engulfed. Here we see a nerve cell, also called a neuron, and obviously it's going to be definitely going to be one of our most active cells. And on the left-hand side, we can see the dendrites going through the soma or cell body. We can see the axon